Yeah, thanks for the opportunity to present our results. I'm presenting here on behalf of the IMPC data cloudification team. And starting with a short description of what IMPC and the data is about. So it's in fact a consortium of mouse phenotyping centers which are located around the globe, which are working on building a comprehensive functional catalog of a mammalian model genome. So the aim is to systematically phenotype over 20,000 knockout mouse strains, in particular one for each gene. Right now, the database has over 8,000 genes phenotyped, and of course, it's continuously growing. And the data is completely freely available and accessible to the whole scientific community. Moreover, the mouse strains generated inside this project are also available for further follow-up research for scientists. And the way how the data is generated is, in fact, a quite complex process and has come a quite long way to standardize this. So there are the 20 different phenotyping centers around the world follow all the same principles to measure the parameters in the different pipelines. They are all well defined in SOPs, so in standard procedures in the so-called IMPRES. And you can see here one example on how the setup looks like. So the mice enter a particular mouse clinic at a certain age, and then the first tests are performed, and then they walk through additional tests after they moved on with the aging, for example, the early adult pipeline, and then continuing until their age of week 58 up to 80 to perform terminal testing and finalized tests. And you can already see that the tests performed inside those clinics are very heterogeneous, and this also relates to the kind of data captured and measured there. And the raw data setup, which you see on the lower plot, is analyzed in a statistical pipeline, and this one brings out so-called genotype-phenotype calls, which means where a significantly detected phenotype is then related to a particular gene which was carried by this knockout mouse strain inside the clinic. And you can see the distribution across the data releases, which is of course continuously growing, and also highlight the challenges which comes with the data, because it's very heterogeneous. It's built up of very different data types, and this data so far was mainly always utilized for presentation.
I was like, okay, it starts from 10, right? How can you come at
there's
like
a background
9:30?
noise.
If you're not speaking,
it starts from 10:04 for me. So
Philip, I'm just going to see if I can track down who that is.
Some questions I was like,
shall I continue? Or? Good.
Please continue. I'm tracking who it is.
Okay, thanks a lot. So yeah, coming again to the point, the data is built up of a very high level of heterogeneity, and this were also one of the main objectives in this project, to go beyond a pure website or simple API accessibility, so to in fact advance this accessibility towards an integration for the analysis of this resource, as well to move it to a portable IMPC data resource. And last but not least, one key point or one key objective is also to enable a so-called user-driven knowledge discovery for the people, so that they are able to utilize this resource in an environment to perform their own analysis of their interest. And to do or to tackle those objectives, the first thing is we were gathering different user queries. So there were in total over 20 different user stories captured related to the IMPC data from various fields, and categorizing them together. It became clear that there are in fact 4 main entry points for people interested in IMPC. So one set of group was always coming with a particular phenotype of interest to query the data. Others were coming with a gene or a particular allele information to access the data. Others were more interested in so-called top-level phenotype category, which is in fact a top-level MP term comprising more significant terms beyond it. So for example, something like nervous system or eye phenotypes. And others more from the mouse community had their main interest in querying the data or accessing it for an experimental parameter or particular procedure, or in fact also a particular data type, for example, imaging data. And to put this all under one umbrella, there was a re-engineering of the of the bulk data access performed, and it is using now a cloud native database, MongoDB, and it has the clear benefit that it's of course available as a service provided by multiple cloud providers. The current API implementation also provides nice queryability through Explorers and REST APIs, and it's also quite easy to maintain and extend. The data organization inside this technical setup is, of course, very user-driven based on the queries I just showed you. So there are mainly two data views centered on genes and phenotypes to provide the particular data bundles. And it also includes all the provenance information and annotations which were accessible through the website so far. That means it captures all the information from the experiments, how the experiments were performed by whom, in which way, and also the annotations from the ontologies. So for particular phenotype, or also the identifiers for a gene or an allele using MGI IDs is all part of this API. Last but not least, one key point was also in regards to the portability to make this available also as a Docker image. So to make it run locally in a, on the one hand, secure environment where this might be necessary, where the people has the need to have it locally directly, but also for scientists who prefer to develop their workflows first in a local environment and then share them at a later point. And in fact, this portability application, this is something we did then as one of the tests. We asked an InfraFrontier node to set the local API endpoint up in their environment, and their summary or their response related to that was very positive. So it's in fact quite lightweight right now and can be set up very easily. And also the queryability was highlighted by them, and also the queries implemented so far provides a lot of combinations of potential use cases there. And what they are now currently doing is then implementing a particular workflow utilizing this data. And of course, one key benefit is here, since they are using this local implementation, once they are on the level to share this workflow, it can be easily adapted to the public instance of this API endpoint. And of course, despite the local portability, the user groups which we also wanted to reach out more, which might not be in the interest of setting it up locally and coming more from the, let's say now, non-mouse community field, to address those people, we also developed a Galaxy tool to make the data, the IMPC data, queryable and accessible inside Galaxy. So there are very different ways to access the IMPC data now through this tool. So you have different entry points like genes, phenotypes, and also the IMPRESS parameters, procedures, and pipelines. One thing to highlight here is also on the phenotype level, we enabled not only to query for mammalian phenotypes, but also for human phenotypes where a particular mapping is available. And one key point also is that this tool is fully workflow applicable, meaning you can in fact use it as a starting point where you simply copy and paste your gene list and perform then follow-up analysis inside Galaxy with additional tools, or you can simply plug it into your existing workflow in the middle or at the end and grasp additional information from your workflow outcomes so far, in addition to the IMPC data. Right now, it's located in the toolshed, and one very next step, which we are aiming for, is also to make this available on public Galaxy instance so that people can use it right away. Now I'm showing you some examples which we did for validation of the tool as well as for the API. So this is, for example, a workflow which we developed already beforehand and used also with other data inside Galaxy, which is performing a gene versus gene comparison. So in fact, the tools which we use here and developed here are comparing the genes against each other and showing you the phenotypic overlap. In the Galaxy implementation of this, you also have a quite dynamic picture of this, meaning if you hover over the different spots, it provides you additional information of what phenotypes are overlapping between those genes. And in fact, this is also quite robust and scalable. So you are able to, in fact, utilize this workflow and this tool with the full set of IMPC genes, so meaning over 8,000, where of course the visualization might not be then the thing you prefer to do, but you can then utilize this data metrics generated here in the background for further analysis. One other example focused on the so-called top-level phenotype category analysis, so where you grasp the information from the via the tool from the API endpoint and categorize the data into those so-called top-level phenotypes, which comprise the more specific phenotypes in the ontology beyond them, and plots this data in a heat map style. Again, in the Galaxy implementation of this is very dynamic in providing additional data to the users. And of course, one clear benefit is once having this in an environment like Galaxy or a workflow management like Galaxy is that you can simply add additional tools. For example, here providing a hierarchical clustering to identify genes sharing a particular phenotypic profile across those top levels. And this now brings me to the conclusion and also to the summary of this. So related to the impact of the project, as we just showed you, so it's now quite easy to perform data analysis based on the user interest, also coming from heterogeneous disciplines. One key point also for InfraFrontier in particular is that we have now an improved and harmonized way of integrating IMPC data in the local nodes, as well as also with our resources, because this comes then into play in advancing the reusability of algorithms developed in the different nodes, since we have now the possibility that people use all the same kind of endpoint and all the same kind of data structure. It's the outcomes of their analysis in terms of machine learning and so on are much better reusable across them and also outside of this community. One key point, in particular on cross-RI integration possibilities, is that with this API endpoint and also with the developments in Galaxy and the portability, it helps us a lot in the OC project, which is currently running, dealing with the integration of different data sets from different research infrastructures. And looking on more on the EOSC level, of course, the two main key things to mention here is that now the IMPC data is accessible through Galaxy, but also that the IMPC data is now in a very well portable state to plug it into different environments. Experience of working within EOSC-LIFE and the technical teams. So we really liked a lot the very supportive environment, which was provided by those various disciplines, so ranging from verification, fair data access, as well as interaction with the people working and developing workflows. And in particular, also the productive engagement with the expert teams was very helpful for us to drive the project into this direction. And the last slide dealing with the future work. One very positive thing is that for us, the sustainability is already clarified between Galaxy and InfraFrontier. So we already decided who will maintain the development of this project also beyond EOSC-LIFE. As mentioned, one very future work next step is the utilization of those developments inside this cross-RI project. But inside InfraFrontier, one of our key focus, particularly on computational developments, is to continuously now tool and workflow development by the EOSC-LIFE members, but also beyond the EOSC-LIFE project. So this is one central thing which is on our agenda. And with this, I want to thank all people who were involved here. So unfortunately, some people needed to leave during the project runtime, and I'm happy to take any questions.
Thank you very much for that really beautiful presentation. I'll declare a vested interest here because members of my group collaborated in this. And I see a question in the chat from Daniela, which says, okay, which is a Zoom question and not about your talk, in which case I will ask my question. So I really like this concept of the bundles, the phenotype bundles and the gene bundles. But I wondered if the bundling on the phenotypes is done by mouse phenotypes or by human phenotypes.
So the bundling on the phenotypes is done by mouse phenotypes. Mm-hmm. Then the access to those bundles is then done by a previous mapping between HP and MP, okay. And it follows the similar principle as it is done for the batch query on the IMPC website. Okay, so my use case that I was thinking was, if I'm interested in all type 2 diabetes related data sets and genes, whether I can easily do that, or whether it's a sort of two-stage process of So right now, it would be that you would need to come with the particular human phenotype. Of course, allowing a disease query is one thing which we are really looking forward to have, because this would enable a whole new user group.
Yeah, and I think there are two challenges with that. So the first is having a set of clear phenotypes from, say, the clinician that wants to ask the question. And then the second is the mapping process, which is quite challenging, just because, we get anatomical differences and differences in assays between human and mouse.
Yeah.
And so I wonder if you are thinking about, what would be a tractable use case for that, and if you've got an idea of where you would start that process and what kind of diseases. Because I know Helmholtz is interested in T2D.
Yeah, so I mean, mapping wise, I would really focus on the let's say on the precise things, because otherwise it's getting too fussy for the user if the user comes with a particular set of phenotypes and does not understand on how the mapping is performed. Of course, this reduces the mapping possibilities a lot. And disease-wise, we are considering currently reusing already work which was done in the Monarch initiative because they did a lot of work there for ontologies and cross-species mapping, and to integrate this into, on the one hand, into the tool and maybe also into the API where possible.
All right, thank you very much. That sounds really exciting. And I think this kind of cross-infrastructure scenario with Galaxy now is really powerful. And I think if we could get the disease thing working, I think that would be really useful. I know certainly that we've got a bit of an underspend on this project on my side, and I think that would be something worth looking at if we can address that, because it's really powerful if we can make it work.
Yeah.
All right. I'm checking in the chat. I do not see any other questions. Could I invite everyone, if they want to ask another question, to Philipp, please? So I'd encourage people to put questions in the chat. Philipp, very nice work. Thank you very much. It's really exciting to see that come to fruition.